Hello again. Welcome to part 2 of Lipid 4 lecture. In this part, we are going to look at a process called lipogenesis, or it also means fatty acid biosynthesis. Lipo is referring to lipid and genesis meaning generation. So lipogenesis collectively means the synthesis of fats or lipids. Lipogenesis is also known as fatty acid synthesis, and sometimes it is referred to as de novo synthesis of fatty acid. Here on these slides, you can see the 5 major steps involved in lipogenesis or fatty acid biosynthesis. We will look into each of these steps in the next upcoming slides. The first step in lipogenesis or fatty acid biosynthesis is production of cytosolic acetyl-CoA and substrate transport. Lipogenesis or fatty acid biosynthesis happens when you consume more calories than what your body needs. So this is when you eat more than what is required. As the consequence of this, you will end up having excessive amount of acetyl-CoA, which is the product of the metabolic processes. Alright, so if you're wondering where does this acetyl-CoA come from, these are the sources. Once you have extra acetyl-CoA inside the mitochondrial matrix, as what you see in the diagram here, your body wants to keep them for future use. And how it's going to keep them? It's going to keep them in form of lipid, to be precise, in form of triacylglycerol or triacylglyceride. So before it converts them into triacylglyceride, it needs to convert this acetyl-CoA into fatty acid chain. So this process is what is known as lipogenesis. Now you have your acetyl-CoA in excessive amount in the mitochondrial matrix. It has to come out from the mitochondrial matrix to the cytosol for the subsequent reactions to take place. But remember that the CoA part cannot cross the inner mitochondrial membrane. So at this point, acetyl-CoA will be condensed with another molecule known as oxaloacetate. This reaction is catalyzed by an enzyme known citrate synthase. So this will form citrate. Citrate then will come out to the cytosol. Once it is out in the cytosol, the citrate will be broken down back to acetyl-CoA and oxaloacetate by an enzyme known as ATP citrate lyase. So now you have your acetyl-CoA ready for the reaction in the cytosol. The second step in production of fatty acid or lipogenesis is formation of methylmalonyl-CoA. So our goal now is to produce this molecule from acetyl-CoA. So here you have acetyl-CoA, and to form methylmalonyl-CoA from acetyl-CoA, we need an enzyme known as acetyl-CoA carboxylase or ACC. So the role of this enzyme is to add the carboxyl group. So the name itself suggests of its action, which is adding carboxyl group to acetyl-CoA. This enzyme has a requirement for coenzyme known as biotin. So basically the biotin is carboxylated, so it picks up the CO2 or the carboxyl group, then it transfers the CO2 group to acetyl-CoA, finally forming the molecule that we want, which is methylmalonyl-CoA. So remember the name of the enzyme here, which is acetyl-CoA carboxylase. All right. So there are certain conditions that promotes the synthesis of melanin CoA or increase the production of melanin CoA. So what conditions are those? This is when you consume a diet containing excess calorie, particularly high carbohydrate diets. Okay, so high carbohydrate diets or high calorie diets will increase the synthesis of this enzyme, acetyl-CoA carboxylase. So when there is increase of this enzyme, there will be increase in fatty acid synthesis because there will be increased production of melanin-CoA, which is the key molecule in fatty acid synthesis. So in contrast, a low calorie or a high fat diet will cause reduction in fatty acid synthesis because this will decrease the synthesis of the enzyme. Alright, and note that the carboxylase enzyme is upregulated by insulin. Alright, so this enzyme will also be uh, produced in high amount when there is high level of insulin. And when do you get high level of insulin? It is when you consume high carbohydrate meal, because that's when the level of glucose in the body increase. So when level of glucose increase, there will be high secretion of insulin. Then the high secretion of insulin will promote the synthesis of the acetyl carboxylase enzyme. So once you have your melanin-CoA, the rest of the reaction involved in fatty acid synthesis is catalyzed by one large polypeptide or enzyme known as fatty acid synthase. So here's the structure of fatty acid synthase. So it is one polypeptide which has 7 different enzymes in it. So look at the structure of it. Pay attention to all the different types of enzyme here. And also it is a homodimer, meaning it is made of 2 monomers and each monomers are identical. And here you have the ACP domain and next to it you have the cysteine domain. Alright, so pay attention to 2 of these sites on this fatty acid synthase enzyme, and I will explain why these sites are important in the upcoming slides. Now recall the structure of fatty acid synthase. Remember I mentioned about 2 important sites in the earlier slide, which is the cysteine and the ACP. Alright, so what happened here? Alright, the enzyme will pick up an acetyl group and put it on the cysteine residue or the cysteine domain. Next, your melanin-CoA will be transferred to the ACP domain, and now your malonyl will be known as malonyl ACP. After that, because these 2 molecules are sitting next to one another on the fatty acid synthase enzyme, reaction now can take place. So this is where you get your reaction 3, where there will be condensation of malonyl ACP with the acetyl group sitting on the cysteine. So ultimately what happens, these 2 molecules will be condensed, forming a 4-chain carbon. Alright, so now you can see how the molecules are being joined together to elongate and form a fatty acid chain. So after that, now you have the 4 carbon chain. Alright, so the subsequent reaction, which is reaction 4, 5 and 6, is aim to saturate this chain with hydrogen atom. So once it has saturated the chain with hydrogen atom, alright, the molecule now will be transferred to the cysteine site or residue of the fatty acid synthase. So now this 4 carbon chain will be sitting on the cysteine residue, and the enzyme will pick another malonyl CoA and join it to this 4 carbon chain. All right, so that is how the process keeps repeating, and every time in the repeats it will add 2 carbon atoms to the chain and elongate the chain until the formation of palmitol ACP, which is 16 carbon chain long. So once it has formed this palmitol ACP, an enzyme known as thioesterase will release the product from fatty acid synthase. Okay, in the earlier slide, we saw a lot of chemical reactions, the structure, and also details. So I just would like to draw your attention to mention that you don't have to remember the structure, the details of the chemical reaction. But what you need to remember is the name of the important enzymes and the name of the important molecules which I mentioned in the lecture. Alright, besides that, you should be able to outline the process of fatty acid biosynthesis. Now, let's look at this slide. In this slide, I'm going to explain about the source of energy for fatty acid synthesis. So, in lipogenesis or fatty acid synthesis, we need energy. Alright, so if you go back to the previous slide, you can see NADPH is indicated at certain stage of the reaction. This means those reactions need NADPH as source of energy. So where does this NADPH comes from? Let's look at the diagram here. So when you consume calorie more than what you need, alright, your glycolytic pathway will take place. Alright, so this is basically carbohydrate metabolism which goes through glycolytic pathway. So in the glycolytic pathway, you produce both acetyl-CoA and oxaloacetate. As we learned before, the extra or excessive amount of acetyl-CoA will be needed for fatty acid synthesis. So here, the oxaloacetate will be fused together with acetyl-CoA, forming citrate, alright, so that it can exit the mitochondrial matrix into the cytosol. Inside the cytosol, the citrate will be broken down, so we get our acetyl-CoA back. Now the acetyl-CoA will go through the reaction that we discussed earlier to eventually form a 16 carbon long fatty acid chain known as palmitate. And this reaction needs energy in form of NADPH, and this comes from the pentose phosphate pathway. So remember, when glycolytic pathway is activated, some of the byproducts from the glycolytic pathway is supplied to the pentose phosphate pathway. So the pentose phosphate pathway will also be activated, therefore supplying energy for fatty acid synthesis, as what you see here in this diagram. Now let's look at the cause of building palmitate, which is 16 carbon chain long fatty acid. Alright, so each malonyl CoA has got 2 carbon. So if you want to make a 16 carbon chain long, you need 7 malonyl CoA, because remember, we fused the first malonyl CoA with an acetyl CoA in the reaction. Alright, so in total you need 7 malonyl CoA. So the malonyl CoA, to make 1 malonyl CoA, we need 1 ATP. Alright, so to make 7 malonyl CoA, we need 7 ATP. Okay, then we need an NADPH. So if you look at the slides, alright, the earlier slides, you can see that NADPH is indicated in two reactions, alright. You don't have to remember in which reaction, but you will need NADPH in two reactions, alright. So if we are joining 7-melanyl-CoA, alright, so this reaction will take place 7 times, alright, and in each reactions we need 2 NADPH, alright. So 7 times 2 will be equivalent to 14 NADPH. So in total we need 14 NADPH. Alright, so the energy equivalent of 1 NADPH to ATP is 2.5. So 14 NADPH is equivalent to 35 ATP when you times 2.5. So the total cost will be 42 ATP. Now recall part 1 of our lecture, which is on beta oxidation. So in beta oxidation, we learned that when we break down a 16 carbon chain long fatty acid, we get a net yield of 129 ATP. Alright, so that is more compared to what we need, alright, compared to the amount of energy or ATP we need to make palmitate, alright. So in fatty acid biosynthesis, alright, at the end you get palmitate, which is 16 carbon chain long. These carbon chain can be fully saturated and can be further elongated or desaturated in a separate enzymatic process, which we are not covering in this lecture. So basically the chain can be elongated further by another enzymes, alright, and these enzymes are found in mitochondria and endoplasmic reticulum. So here's to draw your attention: the human cannot introduce double bond to the chain beyond carbon 9 to 10. So that's the reason why we need to consume polyunsaturated fatty acid, alright, which comes from the diet. So we have double bonds beyond carbon 9 and 10 in the fatty acid chains that we form in our body. So here's the last slide of part 2. So here I have summarized, alright, the differences between synthesis of fatty acid and breakdown of fatty acid, or degradation or beta oxidation, which we have covered in part 1. Note the differences and learn how to compare them. So that's the end of part 2. Thank you very much.